Okay, continuing from where we left regarding the World Health Organization manual on Ebola and Marburg virus disease epidemic manual, we were left on the third stage. The third stage is during the outbreak. What is the response and containment operations? Basically, once the Ebola or Marburg outbreak is confirmed, a multi-sectoral outbreak control strategy is implemented. It involves 4 steps. First, the coordination of prevention and control activities and the mobilization of resources. Second, the creation of a surveillance system. Third, the promotion of a social and behavioral intervention program. And fourth, clinical management of Ebola and Marburg patients in the affected area. Now, the fourth stage is what happens after an outbreak. It is the post-epidemic phase. Basically, once the epidemic is over, surveillance is resumed, just like those that took place in the pre-epidemic phase. Now, our second example is chytridiomycosis. It is actually an important emerging disease in amphibian populations. It is caused by an aquatic chytrid fungus called Batrachochytrium dendrobatidis. It is the causative factor of major global decline in amphibian populations. It is found on every continent where amphibians occur except the Antarctica. Now, a little bit about the history. According to Weldon and others, chytridiomycosis was first registered in 1938 in Xenopus levis frog in southern Africa, where the infection remained a stable endemic disease for 23 years. However, global dissemination then took place, and it actually involved a series of key steps. The first one, the occurrence of the fungus in an amphibian vector, in this case, Xenopus levis, in southern Africa that is relatively resistant to disease. Second, a rise in the international trade of this species for technological advances, such as the Xenopus pregnancy test used in human medicine. This obviously took place in 1935. Third, the escape of the pathogen and establishment of feral populations of Xenopus levis. Fourth, the transmission into naive susceptible hosts. And fifth, further transmission to other countries along different trade routes in key amphibian vectors. Obviously, this moves in a higher numbers and become established in commercial populations that interact closely with wild frogs. Now, these trade routes include the food trade, such as that of the American bullfrog, which is very popular as a food item. The trade for scientific research or laboratory use. We already saw the case of the Xenopus levis. Bait trade, for example, that of the larval eastern tiger salamander, which are exported within the US. The pet trade, and human-mediated translocations, which can be either intentional, for example, that used for biocontrol or the release of exotic species into the wild for aesthetic reasons, unintentional, like that of the transport of amphibians during movement of food products, and those used in conservation programs. Now, the second species of Batrachochytrium, which is Batrachochytrium salamandriferans, was discovered in 2013 in the Netherlands. Now here we can see a diagram telling us the days that it appeared in all the different continents. Now, a little bit about the epidemiology. Well, as I just mentioned, it has 2 etiological agents, both Batrachochytrium dendrobatidis and Batrachochytrium salamandriferans, which is mainly restricted to urodels, which are newts and salamander. It has a broad host range, so it affects anurans, frogs and toads, caudata, salamanders and newts, and caecilians. Now, the pathogenic effects of B. dendrobatidis and the resistant to disease is actually species specific, because some species are highly sensitive and the infection has devastating effects on the population, such is the case for the littoral amphibian species. Other animals appear to be unaffected, but may act as potential environmental reservoirs for the fungus. It actually appears that amphibian populations develop some measure of resistance to the fungus over time, or the fungus becomes less pathogenic, or both, who knows. As frog populations that declined dramatically when the fungus first arrived, now appear to have stabilized and coexist with the fungus. There is no sex or age-linked predisposition, since tadpoles are commonly infected as well, But there is age-linked mortality, as adults and juveniles die from chytodermicosis, while tadpoles have not been reported to die. The prevalence of B. dendrobatis in the wild varies with species, life stage, season, altitude, and to a certain extent, temperature. Now, the incubation period for the disease is between 14 and 17 days post-exposure. Stream-dwelling species at high elevations in Central America and Australia have been shown high susceptibility. They are the most that present population declines. The growth and survival of Batrachochytrium species is actually temperature dependent. For example, the optimal growth of B. dendrobatis is observed between 17 and 25 degrees Celsius, and at pH of 6 and 7. At 10 degrees Celsius or lower, the fungus grows slowly, while at 28 degrees or higher, it ceases to grow completely. For B. salamandrivorans, the optimum temperatures for growth are between 10 and 15 degrees Celsius. It grows slowly at 5 degrees Celsius, while temperatures of 25 degrees and higher are lethal. Climate change indirectly potentiates the spread of the disease. At warmer temperatures, amphibians expand or shift their elevational range upwards, so the fungus is introduced into new areas. Now, regarding the transmission of the disease. The fungus begins as an aquatic spore with a single flagellum. These zoospores disperse within the environment and encounter the thin, permeable skin of amphibians. The zoospores then penetrate the skin and encyst, absorbing their flagellum and forming a cell wall. Subsequently, the cyst germinates and develops a small germ tube, which is used to allow tissue and cell penetration. It proliferates intracellularly, and the germ tube evolves into the sporangium. The infected cells are carried to the skin surface during epidermal differentiation, where the mature zoospores are released back into the environment. This is the step in the process where the highest damage is done to the amphibian skin. Now, it's important to note that zoospores are the primary form of dispersal and are fundamental to amphibian pathogenicity. Tadpole infections can be carried through metamorphosis and cause high mortality in juveniles and adults. And the transmission of the fungus by fomites, for example equipment, is also possible as well. Now, the impact of animals. Well, regarding clinical signs, B. dendrobatis infects the keratin-containing layers of amphibian skin. In tadpoles, it's basically the mouth parts. There is depigmentation and defects of the area. During metamorphosis, the infection can spread to other parts of the body. And in juveniles and adults, it's basically focused on the ventral area, the digits, and the pelvic patch. Okay, what are the clinical signs according to the life stage of the animal? In a neuron larva, there is depigmentation of the mouth parts without mortality. Sublethal effects include lethargy, poor swimming abilities, low foraging, and small body size. In metamorphic neurons, there can be sudden death without obvious disease, or skin changes which are mainly focused on the ventral abdomen, especially the pelvic patch, feet, and toes. However, these skin changes are typically microscopic and not detectable at the clinical level. But if it were the case, they may include abnormal skin shedding, so skin shed more frequently and in smaller amounts, thickened skin, swelling, and erythema. Now, in infection by B. salamandrivorans, there are erosions and extensive epidermal ulcerations, excessive shedding, anorexia, apathy, ataxia, and death. When there are severe infections, there can be anorexia and lethargy, and central nervous system signs, which include slow and uncoordinated movement, abnormal sitting posture with hind legs extended, tetanic spasms, loss of riding reflex, and paralysis. It seems that in the disease there is also impaired respiratory and osmotic regulation, which disrupt the electrolyte transport. The electoral blood levels drop, which leads to cardiac arrest and death. Now, the major impacts of the disease so far is basically, as mentioned before, it is the greatest disease-caused loss of biodiversity in both frogs and salamanders. It has caused the decline of hundreds of amphibians. And actually, according to the IUCN, 32.5% of all amphibian species are threatened, and more than 400 species may be on the brink of extinction due to chytridiomycosis. Over just the past 30 years, the fungus has caused the decline of extinction of at least 200 species of frogs. Species extinctions due to bee dendrobatitis include the sharp-snouted day frog, the northern gastric brooding frog, the southern gastric brooding frog, the southern day frog, and the Costa Rican golden toad. B. salamandrivorans is actually very important as well. The family Salamandridae is highly susceptible. The infection is lethal, with animals succumbing within 2 to 3 weeks after initial exposure. The most affected appear to be fire salamanders, with disease outbreaks in the Netherlands and Belgium having reduced population numbers of these animals. Now, surveillance and control of chytridiomycosis, it depends on whether it's in the wild or in captivity. In the wild, prevention can take place following field hygiene procedures. Clothing, equipment, and vehicles need to be checked, cleaned, disinfected, and put to dry, and doing disease risk analysis for translocation processes. Now, surveillance. According to the OIE Training Manual on Wildlife Diseases and Surveillance, there are 4 steps. The first one is the detection of dead or diseased wild animals and the collection of samples from wild populations. B. Identification of pathogens and diseases for mainly carrying out laboratory tests. C. Information management, so this involves recording, analyzing, mapping, and securing all information. And D. Data analysis and communication of results, which is required in 4 areas of public responsibility: public health, domestic animal health, wildlife conservation and management, and environmental management. Now, diagnosis of chytridiomycosis is based on detecting the fungus in the skin of amphibians by either cytology and histopathology, which is very useful because large numbers of organisms are usually present in sick individuals and can easily be detected this way. The histopathology obviously evaluates the severity of skin lesions and can help determine if the infection is clinically significant. It can also be done by light microscopy. It can be examined the skin without staining or stained. And the third diagnostic method is PCR, which helps quantify the amount of fungal DNA in the sample. It can detect small number of organisms, which is useful for detecting subclinical infection. Both conventional PCR and real-time Taqman PCR are used. Now, in Australia, the chytrid fungus is listed as a key threatening process in Australia by the Commonwealth Department of Environment and Heritage, and a threat abatement plan has taken place. This plan proposes a risk-based approach to be used for chytridiomycosis. Globally, amphibian chytridiomycosis is listed in the OIE Wildlife Disease List and declared an international notifiable disease. Now, in captivity, prevention is primary as well, so quarantine is very important. New animals need to be tested for the fungus prior to their entry into an established collection. It can be diagnosed using histopathology examination of dead animals or PCR testing of skin swab samples from living animals. Now, if animals are declared positive, they need to be treated with antifungals prior to their release from quarantine. Husbandry practices are also important, so the movement of substrated water animals has to be controlled to ensure that no dispersal of infection takes place. And biosecurity measures such as the use of disinfectants is also crucial, especially disinfectants to which the amphibian fungus is susceptible. Now, conclusions. Basically, emerging infectious diseases are on the rise. They are present globally and affect all groups: humans, animals, plants, and the environment. Just like there are cases in which pathogens cause significant disease in wildlife, there are other cases in which wildlife are asymptomatic carriers that may act as a source of infection, especially for naive susceptible populations. This is something that we all have to consider and keep in mind. Both anthropogenic or natural factors, like climate change, for example, intervene in the origin of emerging infectious diseases. And with people and domestic animals coming in closer contact with wild animals, the potential for emerging infectious diseases to appear and or continue spreading is always there. Okay, thank you very much for your attention, everyone, and I hope you enjoyed the presentation. See ya!